So now it's time to start talking about gene expression in some detail. We've talked about transcription and translation, and so we know what gene transcription is, or gene expression is, but we really want to talk about the control of gene expression. So that's probably one of the most important things about genetics. I know I say that a lot, but how genes are regulated and how they're turned on and how they're turned off. So the control of gene expression. And a lot of what we know, like other things, comes from studying prokaryotes. So we're going to, this whole chapter, chapter 16, is about how we learned about gene expression and some of the experiments that were done a long time ago on E. coli to understand how genes are expressed. So this is a picture of E. coli. It's a rod-shaped bacterium. It's a bacillus shape. Take microbiology and you can learn all about it. It's just kind of a cool picture. You can see some of these cells dividing here, but these are bacteria, so they're pretty cool. And so this chapter goes into, it starts out with a little bit of general talk about gene regulation, and then we'll get into some nitty-gritty details of bacterial gene regulation. And a big thing that came out of this, and so this is sort of a really long story to talk about genes and regulatory elements, levels of gene regulation, and DNA binding proteins. This part is mostly in eukaryotes, so we'll come back to this more when we talk about eukaryotes. But genes and regulatory elements and DNA binding proteins are a huge part of this chapter. So you have a gene, could be any kind of gene, eukaryotic, prokaryotic, whatever, and we have some kind of sequences that are near the gene, maybe they're far away from the gene, and these are what we would call regulatory elements. So when you see element, it usually means DNA sequence. Then there's some proteins that bind onto these elements. So these are DNA binding proteins because they bind onto DNA. And so looking at genes and bacteria is how we figured out that these things exist. And then we sort of expanded that to eukaryotes. So that's basically that. So that's kind of an overview of what we're going to be talking about. When people start sort of formally thinking about regulating genes, this is a little bit odd, but a structural gene, so certainly when we're talking about bacteria, we can think about things in terms of structural genes. So those are genes that encode proteins. Regulatory genes, I don't really like this term because this is before people really knew what genes were. They sort of separated things out into structural genes and regulatory genes. But now a regulatory gene, it says, encodes a product that interacts with other sequences and affect the transcription and translation of these sequences. Well, these regulatory genes, well, you know, they might make a protein. So that's a little odd. So I like to think of more about in terms of regulatory elements. And so we'll come back to that. A lot of this formal stuff, I think, is better when we start thinking about specific examples. But here are some terms that are going to be important for understanding bacterial gene expression. Constitutive gene expression. That means continuously expressed. You could actually have something that's constitutively on or constitutively off, but we tend to talk about it as being constitutively on. So always on. So we might have a gene that's always on. You could have something that's constitutively off, and it's always off. But we don't always talk about that. And then we're going to talk about how genes are regulated. Some genes they start out, positive control is that the gene starts out off and then something turns it on. And then there are other genes that are trying to be on, they're on naturally, but there's something that turns it off. And so that's positive control, means something is turning this gene on, and negative control is something that turns the gene off. And we'll come back to that a little bit. Now this is kind of weird that they put it in this chapter. This is sort of taking a step back, looking at gene regulation overall, but this is more suited to looking at gene regulation in eukaryotes. But since they have it here, we'll be talking about, there's probably some of this in eukaryotes a little bit, but we talk a little bit about chromosome structure, where the DNA is compacted, and the genes are off. And if you relax the DNA or loosen it up, then you can transcribe the gene. Then you get a pre-messenger RNA. Then there's mRNA processing, and you might get a 5' cap and a poly A tail. And then depending on some of those modifications of the RNA will have different stability, or it might get transported to the cytoplasm better or worse, or it might get translated more or less efficiently. And then you make a protein. That protein might be inactive, and then there's post-translational modification. So these are lots of levels of gene regulation, but these are, all of these things, this is much more relevant to eukaryotes. So this is just giving you a bigger picture. When we talk about prokaryotic gene regulation, we're just going to be talking about transcription regulation. And for a long time, that's really what people talked about when they talked about gene regulation at all, was at the transcription level. Then after transcription, you have post-transcriptional regulation. And then you can actually have post-translational modification. But this chapter is all about regulating transcription, and it's all about DNA sequences that are near a site of transcription, and then the proteins that bind them. So that whole chapter is about that. So, and we learned about, and that's a general thing, DNA sequences and proteins that bind them. That's common to everything. And if you look at these DNA proteins, you very often find specific DNA binding domains. So a DNA binding domain, and a domain is a region of a protein. So if you have a protein, and then maybe there's a specific region right here that does a specific thing, maybe that's the DNA binding domain. So let's say you have some DNA, and then you have a protein that binds onto that. The part of the DNA, the part of the protein that binds the DNA is the domain, the DNA binding domain. Motif is a structural element. It's something that is recognizable. You can look at the amino acid sequence and say this particular structural motif is usually found in DNA binding domains, that kind of thing. So here's an example of some motifs, and then if you put a few motifs together, you can get a domain. So this is a helix. This is a helix turn helix domain. So that means that there's an alpha helix, and then there's some kind of turn, and then there's another alpha helix. Zinc fingers are a common domain. These are loops of proteins that are sort of held together by a zinc ion, and so that's because the zinc holds that together. It looks like a finger, and very often these zinc fingers will bind DNA, and DNA will kind of be in between these fingers. A leucine zipper. These are two proteins that are held together by a bunch of hydrophobic amino acids. So, for example, leucine. And these things zip together like a zipper and hold the protein together. And then this part binds the DNA, so the DNA goes between there. And so in this case, the leucine zipper actually is a dimerization domain, where it holds the two proteins together. And by putting them together, then this is the part that binds DNA, so that's the DNA binding domain. And so kind of a general thing is that proteins are organized into domains with different parts of the protein doing different things, and you can separate them out into DNA binding domain, dimerization domain, activation domain if you're going to activate gene expression, There's a lot of different domains. You can have an ATP binding domain. These are just sort of common regions of proteins that have known functions. Okay, so I need to pause here for a second. Okay, and so the context of this is you have a DNA sequence, you have a transcription start site, you have some DNA sequence element, and you have a DNA binding protein that binds that. And this particular domain that binds the DNA can have these recognizable sort of motifs. So these are motifs, domains. Those terms are almost, they're kind of similar, but there are recognizable things. If you have an unknown protein sequence and you look at it and it has a helix-turn helix, that suggests, well, I know a lot of proteins with helix-turn helix domains, they bind DNA. So maybe this one binds DNA. So it at least gives you sort of a hypothesis to get started. These are just some specific examples of different types of domains. I wouldn't worry about it too much. When we get into just if you have the idea of specific domains and if you see these domains and think about DNA binding, that's fine. As you get into molecular biology and biochemistry, you'll get into that quite a bit. What this chapter is really about are operons, and that's how bacterial genes are structured. And so this will get into this idea of polycistronic RNAs again, and we'll talk about that. Negative and positive control, and inducible and repressible operons. This is a very confusing part of this chapter, and so we're going to kind of skim over it a little bit, because you can have negative inducible, negative repressible, positive inducible, and positive repressible operons, and it's a little bit weird to keep it all straight. We're going to focus on the lac operon, and it is negative and inducible. And so when we get to it, we'll talk more about what those kinds of things mean. The thing I like to focus on most in this chapter are the mutations. Because this is a genetics course, a lot of what is known about the lac operon has come from the analysis of mutations back in the day with these two guys, Jacob and Monod, or Monod, however you say it. They were instrumental in figuring out this lac operon, and they came up with this whole idea that there are parts of the gene that regulate its expression, and that there are other things that bind onto those things and help regulate it. When we talk about, so for the most part, we talk about the lac operon as being negatively regulated, and it's an inducible operon. There's also another level of regulation of the lac operon where it involves positive control in this process called catabolite repression, and this is the role of glucose in regulating the lac operon, and this is the role of lactose in regulating the lac operon. So we'll have to look at both those things. Trip operon we're not going to talk about. Look it up. You'll probably see it in the launch pad assignment, and it's kind of good to know, but if we're really taking home the take-home message from this chapter, we really want to talk about the lac operon. Okay, so the operon, the idea of an operon is that you have oops, go back there. That's probably good enough. Alright, so the idea of an operon is that you have a bunch of proteins coded for in the same area of the DNA. So each of these is an open reading frame. And these are all related proteins. These all make related proteins that are related in function. It means they all do something. And they all do something, they're all needed to do one thing. So in the case of the lac operon, we need to use these proteins to metabolize lactose. So we need all those proteins at the same time. So the gene for all those proteins is controlled by the same elements. So we have some kind of regulatory sequence elements that regulate whether or not this gene is on or off. And if it's on, all three genes are expressed, or all three proteins are expressed, and if it's off, all three are not. So that's the idea of the operon, and we'll look at the specific sequences in a minute. And then there are other genes. So in this context, these are the structural genes. So these make the proteins that are needed for whatever biochemical process we're talking about. And then these are the regulatory elements. And then somewhere else, far away, is a regulatory gene. And so this makes a protein, and then this protein will bind onto some of these regulatory elements and control expression. So this is a general picture of operon structure. We have structural genes. Once we transcribe this, so if we turn this gene on and we make an RNA, then each of these is an open reading frame, and each one will make a protein. The protein will be different, so the protein will be different, but each of these things will be involved in the same process. All right, so it's going to make more sense when we talk about the lac operon specifically. So let's do that. All right, so this is a general, well, we're not quite ready for the lac operon yet. So this is a general thing. So this is showing your regulatory gene over here. And it has its own promoter, and it gets transcribed and translated and makes a protein. And then that protein binds to a sequence on some other set of genes called an operon. And this regulator protein, so all of these operons are going to have some regulatory sequences, and they need to have a promoter. And a promoter is always the same thing in every situation ever. In terms of gene expression, a promoter is where RNA polymerase binds. So you need to transcribe the gene, you need RNA polymerase to bind and transcribe the gene. And then the next sequence is called the operator. And an operator always is bound by a repressor. So the protein is the repressor, the DNA sequence is the operator, and in the other case, the promoter is the DNA sequence and the RNA polymerase is the protein. And so we have, again, we always have a DNA sequence, sometimes called a DNA sequence element, and then we have a protein that binds it. So that's the fundamental thing about gene regulation. Genes have specific sequences that control whether they're on or off, and then they have proteins that bind those sequences and either turn the gene on or turn the gene off. Anything else here? Oh, well, so the other thing that's interesting here is, so we've got these 3 open reading frames in one RNA. So what do we call that? It's a polycistronic RNA. It codes for several proteins. Now each of these proteins makes an enzyme, and that enzyme, each of these enzymes is somehow related in the same biochemical pathway. So they're either making something or breaking something down, but they're all needed. So in the lactose operon, all of those proteins are used to metabolize lactose for energy. In the tryptophan operon, all the proteins are used to make tryptophan. And so whatever the process is, you need all those proteins to do it. And so when you need one of those proteins, you need all of them. And so you just turn them all on or turn them all off at the same time. So this would be a good little exercise for you. maybe pause it and draw your own little general structure of an operon. And what you should have is a transcription start site, some structural genes. Each of these structural genes has its own open reading frame. So make that a TGA, another open reading frame here, maybe that's a TAA, another open reading frame here, maybe that's a TAG. Those are the three different stop codons that you can have. And then you need to have some regulatory sequences, and you need to have a promoter and an operator. And the operator is always between the promoter and the transcription start site, because the operator is an on-off switch. And so it's so the RNA polymerase may or may not bind to the promoter, and if it's bound, it depends on whether or not the repressor is bound to the operator. So always put the operator in between the transcription start site and so this is the transcription start site, and the promoter. And that's specific to bacteria. So this is now where operons are specific to prokaryotes. And then somewhere else, far away, there's another gene. It has its own little promoter, and it gets transcribed and translated and makes a protein. The example that we'll use, that protein is a repressor, and that repressor binds the operator. So I think that's it. The key is that these guys are all three of these, so these are the structural genes. And these structural genes are all regulated by the same regulatory sequences. So they're all either on at the same time or off at the same time. So inducible or repressible operons. Okay. So the best way to illustrate this, so first we'll do inducible. And with inducible, and this is gene expression. And with inducible ones, you start out with the genes being off. So this is low gene expression. You add something, and the gene gets turned on. So we say that that gene is induced, and the thing that induced it is called the inducer. Seems reasonable. A repressible operon is a gene that starts out always being on. Then you add something, and that turns it off. And so this thing that you add ends up being called the co-repressor. It says repressor, R-E-P-R-E-S-S-O-R. But the main thing is the shape of the graph. Inducer, you add something, it turns it on. Repressor, you add something, it turns it off. That's basically what it is. Now, when we get to exactly how the proteins work, this is where the positive and negative comes in, and it gets confusing. This seems positive. When you induce something, it seems positive. We're going to see, but an inducible operon can be positive or can be regulated by a positive or negative mechanism. A repressible operon can be regulated by a positive or negative mechanism. So you can have 4 different kinds of things. We're going to focus on lactose, and lactose is one of these. The lac operon is one of those. So positive inducible operon, I thought I took this out. So the positive or negative has to do with the regulatory sequence. So you can have a regulatory sequence that controls a gene. If it's bound by a repressor always turns genes off. It represses gene expression. Repressor turns genes off. And so that is negative. It's turning the gene off, so it's a negative mechanism of regulation. Positive regulation has a DNA sequence, and there's a protein that binds it, but that protein is called an activator. So when we're talking about positive or negative, we're talking about whether or not the protein that involved turns the gene on or turns the gene off. So this turns the gene on and it'll get transcribed. A repressor turns the gene off and it won't be transcribed. And then the inducible or repressibleness is irrelevant really when it comes to that. So we'll talk about, we're going to talk about the lac operon, and it's going to be, the lac operon is going to be negative because it's controlled by a repressor and it's inducible. And why don't we just focus on that, and if we have time we'll look at some other examples. So the lac operon is a negative, I probably should have just taken that last slide out, but a negative inducible operon, we'll show what that means in a minute. It's involved in lactose metabolism, and lactose metabolism means you take lactose and you break it down into glucose plus galactose. So now you can use it for energy. You can do glycolysis, Krebs cycle, all that stuff, and get some ATP. So you only need these genes, so the genes are involved in this metabolic process. And so you only need them when there's lactose around. If you don't have any lactose, there's no reason to express these genes. And also, if you already have glucose, you don't need to express these genes because there's no sense in making more glucose if you've already got some. So we'll talk about how that's regulated. And these are some of the key features of the lac operon that we'll be talking about. So we have the structural genes are down here, and these are the genes that make proteins that are involved in this process somehow. And then you have lac I encodes the repressor, lac P is the promoter, and lac O is the operator. Now, see this little I right here? That is not the inducer. That is the repressor. I would like to call it lac R, and in some other operons they do call it R, but they call it I don't know, for whatever reason. Okay, so the structural genes are lac Z, lac Y, and lac A. Lac Z encodes beta-galactosidase, and that's the enzyme that breaks down lactose into glucose plus galactose. So this enzyme is beta-gal, or beta-galactosidase, and it's encoded by the lac Z gene. Lac Y encodes a permease, so if you have a little bit of lactose out here, a little bit of this lactose can get into the cell. That turns on this gene, or this operon, and expresses those proteins. One of those proteins, the one in the middle here, goes to the cell surface and sort of pokes a hole in the membrane so that more glucose can get in. And so it's going to make a bunch of proteins that make these pores in the membrane so that the more glucose can get in. So the idea is that the cell is sensing that it's in lactose, and it says, okay, well, let's bring that lactose in, and then once it gets in, beta-galactosidase breaks it down. Don't worry about this one. That is some kind of obscure biochemical process that is not worth the effort to explain. All right, so let's draw the lac operon. So we know a general operon has a transcription start site. It should have a promoter and an operator. And if we're talking about lac, this is the lac promoter and the lac operator. Then it has 3 structural genes, which are lac Z, lac Y, and lac A. And then somewhere down here, it has lac I. What do these things do? This makes beta-galactosidase. This makes permease. And this one makes transacetylase, but we don't care what that one does. Well, it's a transacetylase, so it transfers an acetyl group from something to something else, but we're not going to worry about that too much. Okay. And the lac I gene makes the repressor. And the repressor binds the operator. RNA polymerase binds the promoter. And that's about it for now. But get used to drawing that. Let's draw real quick. P O Z Y A, and down here we have I. So we often write this like this. If we want to just quickly represent all elements of the lac operon, we write IPOZYA. Alrighty. Alright, so this is just showing a close-up of what these proteins do. And so this only happens, so we only need the lac operon when there's lactose in the environment. And so here's some lactose, and this is, so each of these little molecules looks like a sugar. And then what we're going to do is break this little bond right here. And so the first thing that happens is beta-galactosidase is going to break down lactose into galactose and glucose. It also changes it a little bit. I don't even know. This is just some subtle biochemical change. And it turns it into allolactose. And so we'll a lot of times we'll just sort of interchange and talk about lactose and allolactose as being the same thing. But you'll see this term allolactose in a minute. And so that's basically it. They don't even mention transacetylase here, but here's your permease protein that's letting lactose into the cell, and then beta-galactosidase breaks it down into galactose and glucose. So that's pretty complicated. So let's back up a second. Alright, let's see. So before we get into that complicated slide, we want to talk about the fact that we only need the gene to be expressed when there's lactose around. So when there's no lactose, the repressor, make the repressor red because it's turning it off. So repressor is bound to the operator. So no matter what else is going on, the gene is off, right? Because the operator is blocking it. And the RNA polymerase may or may not be bound to the promoter. But even if it is bound to the promoter, it can't go this way. It can't get past the repressor, so it's turned off. Then when you add lactose, lactose binds onto this repressor. And so let's say this is lactose, and it's actually allolactose that does this, but I don't care. So call it lactose, call it L-lactose, whatever you want. It binds onto the repressor, causes the repressor to let go of the operator. Then if the RNA polymerase is bound to the promoter, then the RNA polymerase can cruise along and transcribe the gene. And as it transcribes the gene, we'll just sort of tie this together a little bit with what we know from before. We'll put the ribosome on there. And because we have coupled transcription and translation, the ribosome hops on and starts making a protein as soon as it can. And so, you know, and this guy's been starting a little bit longer, and it's making the protein. So just to kind of put all that together. Then it's going to make all three different proteins. So you're going to have three different open reading frames, and the ribosome sort of cruises by one open reading frame, releases that protein, then goes and transcribes the next gene, and releases that protein, etc. But the key is that in the presence of lactose, the gene is turned on, and it's turned on because the lactose is binding the repressor. The repressor lets go of the operator, and that's what happens. And so this is explaining this inducible characteristic of the operon. So it's usually off, so no lactose, the gene is off. Add lactose, and the gene is on. And we're measuring gene expression. You can measure gene expression in various ways. Who cares how we do it? Usually we measure beta-galactosidase activity. Let's see. So we add lactose, gets turned on, and so what happens right here? Well, what's happening right there is lactose is binding on to the repressor. RNA polymerase is present, and it starts transcribing the gene, and then you make the proteins, and you're off to the races. So because lactose is turning this gene on, we said before that this was an inducible situation, so lactose is the inducer of the lac operon. And inducers kind of always have the same effect. An inducer is something where you add it and it turns the gene on. That's the definition of an inducer. And then this is kind of a complicated view of what we just showed here. And so down here is all your structural genes. Oops. So your structural genes are down here. Your operators here, your promoters here. And so this is in the absence of lactose, the repressor is bound. At the moment there's no RNA polymerase on there. But RNA polymerase can bind if the repressor is there a little bit. And then here we've got, you can see this little purple dot here is lactose, or allolactose. It's a very minor modification of lactose. It binds the repressor lets go, and then we get transcription and translation, and then we can convert lactose into glucose and galactose. So that's what's going on there. That's like the most complicated diagram. So that's why it's good for you to just draw them out yourself. Lac operon in the absence of glucose and the absence of lactose. Now we haven't talked about glucose yet, so don't worry about that. But in the absence of glucose, RNA polymerase combined. If we've got lactose involved, lactose binds onto the repressor. The repressor lets go. RNA polymerase cruises along, transcribes the gene. Ribosome comes along, makes protein. the proteins fold up into their three-dimensional shape, and they do what they're supposed to do, such as converting lactose into glucose plus galactose, or going to the cell membrane and making it permeable to lactose. So I would get used to drawing those out. Even if you are answering questions that are not involved with, where you're not asked specifically to draw it out, if you see a stretch of questions that are involved in lactose metabolism or the lactose operon, and you start drawing these things out, then it helps you answer the questions. It's very difficult to answer these questions just sort of off the top of your head. Always have a, this was supposed to be the absence of lactose. My bad. So this is in the presence of lactose, sorry. So we'll switch them up. Presence of lactose. So the presence of lactose, you have to have lactose there, so it turns the gene on. So the gene is on. And by the way, this is the only time the gene is on. In the absence of glucose and the presence of lactose. Gene is on. In the absence of glucose and the presence of lactose, again, don't worry about glucose yet. Just know that with glucose, the RNA polymerase can bind the promoter. So this is the promoter, and this is RNA polymerase. Absence of lactose, the operator is bound by the repressor, and the gene is off. That is in the absence of lactose. There's no lactose there. And then lactose comes in, so we'll just do presence of lactose on the same page. Presence, so we add some lactose, and we know that if we add lactose, the gene's going to be expressed. How is it expressed? The lactose binds the repressor, repressor lets go, RNA polymerase cruises along, transcribes the gene. So presence of lactose. So at one level, you just need to know this. You just need to know what happens in the presence and absence of lactose. And know all this terminology that goes along with it. This is a close-up of what the promoter and the operator look like. And so this is just to sort of reinforce some things we talked about before. A promoter has this -10 and -35 sequence that's very common in bacterial promoters. There's a transcription start site. And here we actually have the operator is overlapping the transcription start site. Okay, I tend to draw it like this with the operator here, but this is just saying it overlaps the transcription start site a little bit. So that's okay. And then your LAC-Z gene starts. And the idea is that there's a specific DNA sequence. And so the proteins that bind these things are DNA sequence specific binding proteins. They don't bind all DNA. They only bind a specific sequence. And this reinforces the idea that the DNA sequence is just information. So this particular sequence carries specific information, and this specific sequence carries specific information, and that information allows RNA polymerase to bind. If you think back to RNA polymerase, maybe the sigma subunit is helping it bind that area. And then the LAC repressor binds this particular sequence. And so the information in the DNA indicates what sequence should be bound. So the presence of allolactose, the LAC repressor, binds the operator? I don't know. Let's draw it out. We got a promoter, we got an operator, then now we know overlaps that. Presence of lactose, the repressor, lactose is bound there, so it cannot. Let's see if we got it right. Got it right. Alright, now we're going to talk about some of these mutations. And then later we'll come back and talk about the role of glucose. So right now, everything we talk about is in the absence of glucose, because you only need this gene on when glucose is absent. If we add glucose, it's going to turn the gene off, regardless of what's happening with lactose. Because the whole point of this gene is to make glucose. If you already have a bunch of glucose, there's no reason to transcribe this gene. So now we're going to back off. We're going to say, so for the rest of this time until I say otherwise, no glucose. And then we're going to look at some of these mutations that Jacob and Monod used to figure out all this stuff. So everything that we just talked about was figured out by these two guys back in the 40s, before they even knew about DNA or genes or anything. They basically just used mutations and figured out this whole system. And some of the mutations that we'll talk about are, so bacteria are usually haploid, meaning there's only one copy of everything. But you can experimentally induce what's called a partial diploid, and you can put in a piece of DNA in addition to the normal chromosome. So if you have a bacterial cell, you have a normal chromosome, you can artificially put in a second chromosome, or at least a little bit of a chromosome. And so then you're diploid just for this region that is covered by that little stretch of DNA. So we make a partial diploid, and you need something to be diploid if you're going to ask questions about is something dominant or recessive, or other genetic questions like that. So we can make a partial diploid. We put it on this thing called an F plasmid. Don't worry about it. All right. And structural gene mutations, I mean, you can mutate the LAC-Z, LAC-Y, LAC-A genes, and they just knock that gene out, so it won't work. But then what we're going to be mostly interested in are mutations that affect the regulatory components, because we're interested in gene regulation. So mainly operator mutations and LAC-I mutations that affect the repressor. And so this is what a partial diploid genotype looks like. So when you see this, the important thing here is this little slash right here. These are kind of hard to interpret sometimes. This is saying one piece of DNA has the LAC-Z gene as wild type and the LAC-Y gene is mutant. But in that same cell, there's another piece of DNA, and maybe the Z gene is mutated and the Y gene is normal. And the reason that we use these things is that if we see any LAC-Z protein, so we can measure the activity of LAC-Z or beta-galactosidase, we know it came from here. And if we measure LAC-Y somehow, we measure the permease, we know it came from here. And so that kind of helps us out to help us answer some questions. Alright, so what's going on here? So what we want to do is we want to look at each of these mutants, and we want to ask whether or not certain genes are expressed. So we're going to come up with these tables, and we're kind of starting out with a more complicated one. These are structural genes. Oops. Lean down a button. Alright, so this is a mutation where we've got this genotype. So we've got Z plus Y minus over Z minus Y plus. Everything else is wild type. And so we'll say that we want to measure beta-galactosidase and permease. And we want to do this in the absence of lactose and in the presence of lactose. So now in this case, in the absence, they're both going to be off because the repressor and everything are working and turning the gene off. But in the presence of lactose, so this is absence of inducer or lactose, so we'll just say lactose for now. These guys are both going to be on. And so what this is telling you is it doesn't matter which side of this line the thing is on, each one of them is going to be expressed normally. So that's just kind of a general way to the kind of table that we're going to look at, and we're going to look at a lot of examples. Then we're going to look at some regulatory gene mutations. Let me go back up here. But if we looked at each of these mutants by itself, let's say we had just a Y minus mutant, and we didn't have the Z plus on the other chromosome. It was just a haploid, and the only copy we had was we have a Z plus, but we don't have a functional Y gene. Well, beta-galactosidase will be off in the absence of lactose. Permease is going to be off in the absence of lactose. Beta-gal is going to be on, so we'll just say plus for being on. But Y minus is still going to be minus because it's not there. So this gene, whether it's expressed or not, we can't tell because it's mutated and we can't measure it. And then if we just had this by itself, then they'll still both be off, or you won't be able to measure them in the absence of lactose. Beta-galactosidase is mutant, so you won't have anything ever, and this will be normal, and it'll be expressed the way it's supposed to be expressed. And so this is just a way to be able to tell where is this protein coming from. We don't know where it's coming from yet, but we can manipulate this, and we can put it by itself, or we can put it on different pieces of DNA, and we can see what's happening. Then we have some regulatory gene mutations, and the most important ones are probably these things that lead to constitutive expression of the genes. All right. Yeah, and the way I think the way they describe this is a little goofy. So we might need a couple slides for this. All right, let's see. So now we want to explain these constitutive mutants. And the first type of constitutive mutant, and again, constitutive means that the gene's on all the time. The first one is LAC-I minus. So if we have a LAC, I keep leaning on buttons, I apologize. So if we have a LAC, so now we're going to add to this story. So we're going to have an I minus. So I'm just going to go ahead and put everything in here. So we're going to have an I minus, we're going to have a wild type promoter, we're going to have a wild type operator, wild type Z plus, wild type Y plus. We're not going to worry about A because we're not going to measure that one. And then we can measure beta-galactosidase or permease. And we can measure it in the absence or presence of lactose. Now, if we don't have the repressor, so the repressor's job is to bind the operator and turn the gene off. So if you don't have the repressor, the gene's going to be on all the time. So even in the absence of lactose, the gene will be on. Even here. So this is the phenotype. When you see everything on all the time, that's what we mean by constitutive. Expression. So what's going on? We have an operator, promoter, operator. Down here we have an I gene. Normally makes a repressor, normally binds the operator, but if it's minus, we don't have this. So that means as long as the RNA polymerase can bind here, the repressor can never bind the operator and the gene's always on. Then it says that LAC-I plus is dominant over LAC-I minus and is transacting. So this is where we're going to use this kind of look, this kind of genotype to sort of look at that. So we got LAC-I plus, and we got the Z minus on one side, and we got the Z plus on the other. So we'll look at a couple of those examples. So first of all, it says that LAC-I plus is dominant over LAC-I minus. Alright, well, if it's going to be dominant, that means we have to have a diploid. So we need to have I plus and I minus. And the way, this is just straight up genetic dominance, is that if you make something heterozygous, whatever phenotype you get out of that, that is the thing that's dominant. And we know that the phenotype of I plus is regulated expression or inducible expression. I mean, it goes on and off. And we know that the phenotype of I minus is constitutive expression. So if we put these things together and we see inducible expression, then I plus is dominant. If we put them together and we get constitutive expression, then I minus is dominant. So we can do one of these deals where we're going to have a normal promoter and a normal operator. And let's do one of these deals where we put the Z plus on one side and we put the Y plus on the other side. And what do we get here? Beta-gal permease, beta-gal permease, and we'll do let's make this minus lactose and plus lactose. So what's going to happen? So I plus makes a repressor. So that's what this guy does. He makes a repressor. If he makes a repressor, that repressor can bind the operator. It's going to make a repressor here and it can bind that operator. So this operator controls Z plus, and this operator is controlling Y plus. But the repressor can bind to either operator. So what's going to happen is both of these guys are going to now be regulated the way they're supposed to be. And we call that inducible expression, where it's off in the absence of lactose and on in the presence of lactose. And that's because this I gene is making this protein, and the protein can diffuse around in the cell and it can bind onto either operator. We call that acting in trans. Because I plus can regulate this operator, and it can go across the line and regulate something on a different piece of DNA, that's what we mean by acting in trans. Trans means across. So if you go across the ocean, you make a transatlantic voyage. I put in a lot of extra slides there. Alright, so this is what's going on with this. We have a wild type repressor, and this is showing here that it's acting in trans. It's acting on that operator and it's acting on that operator. If it can only regulate one operator, we call that cis. So the difference between so we're talking about cis versus trans. But this example is acting in cis. Now you might, if you've had organic chemistry yet, you might remember that this is cis and this is trans. So cis and trans are very generic words. And so trans means across, because those hydrogens are across the bond. It's called trans, and cis means they're on the same side. Even now, these days, you hear of cisgender and transgender. And cis means the same, that your psychological gender and your genetic gender are in line. And trans means that they're different, that they're sort of at cross purposes, sort of. So cis and trans just generally mean same and different. Yeah, I think we did this one already. Alright, so now this one's talking about a super repressor. And it's kind of weird. They just kind of throw them all out there. I'm going to go over a worksheet that has all of these things together. Alright, so a super repressor is actually kind of a cool one. So we have, I find it hard to read these diagrams, so I'd rather just draw it myself. So a super repressor is a repressor that binds the operator. So there's a repressor. But, so here's lactose. Lactose cannot bind onto the repressor. So the repressor is always bound. So here's your super repressor. The super repressor binds onto the operator, and binds onto that operator, and once it gets bound, it gets stuck. So even if there's normal repressor around, this repressor will bind onto the operator and bind on there and get stuck, and it will prevent transcription from happening. So that's a super repressor. So if we have this genotype, so in this little table that we're going to do, we have a super repressor, normal promoter, normal operator, normal Z, put it over a normal repressor, normal promoter, normal operator. This one has it over a normal Z. And we can put Y there too. And it doesn't really matter. Everything's going to be off. Anytime you have a super repressor, unless there's something else going on, then everything's off. And so we'll have minus lactose, plus lactose, for measuring beta-gal or permease. The super repressor represses all the time. So it's super. It's a big time repressor, represses everything all the time. We have some other constitutive mutants. So these are, so any mutations that are in, so when we get to this, so I mutations, or repressor mutations, act in trans. So this can get pretty complicated, but if you just remember that all repressor mutations act in trans, then you're good. Operator mutations act in cis. Alright, so these things are cis acting. And so just to draw this out, a lot of the symbolism is a little bit weird. All we're saying is that if we've got an operator here and an operator here. So an OC is a constitutive mutant. So let's put Z plus here and Z minus here, and Y plus here and Y minus here. Alright, well hold on, let's just do it. Let me make sure I have it the same way they have it. Alright, so if we write out the full genotype, so we're going to have I plus, O plus, Z minus, so that's this. We're going to put that over I plus, oh, we got a normal promoter, normal this. Put Z plus down here, but now we're going to put this constitutive operator next to Z. And I'm going to make it a little bit better, and we're going to put Y plus up here and Y minus down here. And so this Z plus is if we have O plus, this normal operator, so the normal operator is an on-off switch. So if the normal operator is functioning, then the Z would go on and off, and the Y would go on and off. The constitutive is a switch that's always stuck in the on position. And so the question is, does this OC or the O plus, do they act in cis or trans? If it acts in cis, then the OC will only affect Z, and and the O plus will only affect Y. If it acts in trans, then maybe OC could come over here and affect Y, and O plus could come over here and affect Z. So we do the experiment, and we ask in the absence of lactose, in the presence of lactose, and we look at beta-gal and permease, what's going on? And we're looking at this overall situation. Well, it turns out that the operator acts in cis. So the normal operator is going to control just the permease gene, and it's going to act normally because it's a wild-type normal operator. But Z plus is under control of the constitutive operator, and so it's always on. This takes some getting used to. You're going to have to practice. And then this just clears it up perfectly. Holy moly. There's a lot going on there. Alright, so what is this thing? This is everything. Now we've got constitutive operators on both things. And what do we got going on down here? A wild type operator. Alright, so this is a little too much. Alright, so this is showing here. This is the partial diploid that has a constitutive operator, and Z is under control of the constitutive operator there, and it's under control of the normal operator here. So but the only Z you can measure, the only beta-galactosidase you can measure has to come from here. And so if it's under control of the constitutive operator, it should be on all the time. And it's on here, and it's on here. And it's on in the absence of lactose, it's on in the presence of lactose. Why is that? So a constitutive operator is one where the repressor can't bind it. That's not a 1. That's showing a line with another line here saying that repressor can't bind the operator. And this repressor can't bind the operator. Well, this repressor could bind the operator, but lactose is binding it. But down here, the repressor, even if you had so lactose is kind of irrelevant in this case, because the repressor just can't bind the constitutive operator no matter what, because the DNA sequence of the constitutive operator is changed, and so the protein doesn't recognize it and it won't let it bind. Over here, we've got the constitutive operator controlling the mutant beta-galactosidase, so you don't get any functional beta-galactosidase ever from the constitutive thing, but you do get it, because now your beta-galactosidase, your functional beta-galactosidase is connected to the wild-type operator, and so this wild-type LAC-Z will be inducible. So it'll be off in the absence, and it'll be on in the presence of lactose. It's a complicated diagram. We're going to go over that worksheet and look at all these different combinations again. So that's kind of what's going on here. If we have our I plus normal promoter, normal operator, mutant LAC-Z. But down here we've got normal repressor, normal promoter, normal operator, wild type Z. Actually, this is constitutive because it's this. So this constitutive operator is going to control this. And if we have minus lactose plus lactose, beta-gal permease, beta-gal permease. The Z plus, the only one that's functional, is under control of a constitutive operator, and so it's on all the time. We don't have the Y involved in here, so I guess I didn't have to put that on there. But because the wild type normal Z plus is under control of the constitutive operator, it's always on. if we put that Z plus under control of a normal operator, it's going to function normally. So this constitutive operator can't go up here and turn that on all the time. So this wild type operator controls that. So it can only control the thing that it's physically connected to. And so it'll be off in the absence of lactose and on in the presence, just like it's supposed to be. So it's inducible. So inducible is the normal phenotype, and this is constitutive. These are pretty simple. If you have a promoter mutation, then RNA polymerase can't bind. And if RNA polymerase can't bind, you can't transcribe the gene. It doesn't matter what's going on anywhere else. And it's cis-acting, meaning if you have something like this, everything else is normal. So this guy, so let's switch this around. Let's put Z minus up here, or Y minus up there, and Z minus down here. Now the Z plus is under control of this promoter, so it's always off because it can't ever be expressed because it doesn't have a promoter. But Y plus is under control of a wild type normal promoter, and so it'll just be normal. It'll be inducible because it's got everything else is normal that's controlling the expression of this. Alright, so we're going to go do that lac operon worksheet, and so we'll come back to that in a minute. But quickly we're going to talk about, so that's the end of that, and that's the main part of the lac operon is what happens in the absence of glucose. And now this is getting into what happens in the presence of glucose. What's the role of glucose in regulating this? So this is just going to be a short little bit. And the worksheet that we do is going to be all in the absence of glucose and looking at how lactose controls it. But here we're going to talk about catabolite repression. And what that means is, so metabolism is all these biochemical pathways in cells. Lactose metabolism is breaking lactose down into glucose plus galactose. And metabolism is you have catabolism, which is breaking things down. So this metabolic pathway is a catabolic pathway. And just to remind you, the other type of pathway is anabolic. So if we're building something up, it's called anabolic. Those are the two types of metabolism: anabolic metabolism and catabolic metabolism. So glucose is a catabolite. It's the result of a catabolic pathway. And there's an enzyme that is carrying this out. And oftentimes there are feedback pathways where the result of a pathway comes back and inhibits the enzyme. So you're probably used to thinking of that, feedback inhibition, where the product of an enzymatic reaction inhibits an enzyme that's responsible for that reaction. But now, rather than inhibiting the enzyme itself, we're going to go back and inhibit the gene that makes the enzyme. So it's the same thing. It's like a feedback pathway, but it goes back and inhibits the expression of the protein rather than the activity of the protein. And it's regulated by this small molecule called cyclic AMP. And so cyclic AMP is a molecule. It comes from ATP and it gets converted into cyclic AMP. The enzyme that does this is called adenylyl cyclase. And glucose, just to make this, just to give you, we'll come into the mechanism here in a minute, but this is needed to express the lac operon. And I'll show you in a minute why. So what does glucose do? Glucose inhibits this enzyme. And so if you have high glucose, you have low cyclic AMP, and you have low lac expression. Alright, so high levels of glucose inhibit this adenylyl cyclase enzyme. That results in reduced cyclic AMP. You need cyclic AMP to transcribe the gene. And so if you don't have cyclic AMP, you get less of the gene. And it looks just like this, which is too complicated. So I'm going to give you the simple version. And then you can come and look at this and figure it out yourself. There's just too much info in there. So what's going on? So you got your lac operon. Got a promoter. Got an operator. Got a repressor down here somewhere, or a gene to make the repressor. And then we also have RNA polymerase. And RNA polymerase wants to bind this promoter. But it's actually a little bit more complicated than that. There's another protein, well, let's make it red, called CAP. And this one's RNA polymerase. We need both of these guys to bind the promoter. So RNA polymerase, well, an RNA polymerase probably binds over here, and CAP binds over here. And now RNA polymerase can transcribe the gene. But CAP can only bind the gene if it's bound to cyclic AMP. So you need cyclic AMP to bind to CAP. Then CAP can help RNA polymerase bind to the promoter. So CAP is actually an activator protein because it's helping to activate transcription, because the function of the CAP protein is to turn on the gene. But it can only do that if there's cyclic AMP. Cyclic AMP comes from ATP, which is made by this enzyme adenylyl cyclase. The function of all these guys when we make these proteins is to take lactose and turn it into glucose plus galactose. So now if we get a lot of glucose, it goes up here and turns off that enzyme. And then you don't have cyclic AMP, and then the CAP protein can't bind the promoter. If the CAP protein can't bind the promoter, RNA polymerase can't bind the promoter. And voila, the gene is off. Alright, so I just did that, but let's do it again. And so you guys should practice. Alright, so let's so the glucose regulation is sort of independent of lactose. So we'll do it in the presence of glucose. So plus glucose minus glucose. And glucose only affects the RNA polymerase. So in the presence of glucose, the RNA polymerase is not bound to the promoter. Why? Because the ATP is not being converted to cyclic AMP. Why? Because glucose inhibits. So this little symbol means inhibits. No cyclic AMP, no cap protein, so the cap protein can't work, and RNA polymerase can't work. Minus glucose, ATP gets converted to cyclic AMP. Cyclic AMP binds onto cap. Cap binds to promoter. Now RNA polymerase can bind the promoter. And now we can transcribe the gene. But whether or not we actually transcribe the gene now depends on lactose. So this is sort of a minimal thing that you need. So up in the presence of glucose, the gene's off. Whether it's minus lactose, plus lactose, doesn't matter, the gene's off. Now in the absence of glucose, if minus lactose, the gene is on, minus lactose, the gene is off, plus lactose, the gene is on. So everything we've talked about so far in terms of how lactose affects the operon, all that's only true in the absence of glucose. So the situation where the gene is actually turned on is here. If we have plus lactose, you got lactose bound on to the repressor. The repressor can't bind the operator. RNA polymerase can bind the promoter because it has cap. And so then we'll transcribe this gene, make the proteins that we need, until eventually we make enough glucose, and then we'll turn it off again. Alright, so that's pretty much it for the lac operon. This is just a picture of the cap protein, so it's binding DNA. So this blue part is the cap protein complex, and you can see some cyclic AMP in there. The cyclic AMP changes the configuration of the protein so it can bind DNA. It kind of bends the DNA. A lot of these proteins bend DNA, and then somehow that helps RNA polymerase hop on there and bind, and then you can transcribe. As long as your operator is unoccupied by the repressor. It's kind of a cool picture. What is the effect of high levels of glucose on lac operon? High levels of glucose. Very little transcription. You might be able to get a little bit of transcription. Most of these things are not absolute. Trip operon, we're going to stop there. I think limited time, you got the general idea of a you got a general idea of how these different things work, or how the lac operon works, which is a good example of the lac operon. There's 50 different kinds of operons, but we'll stop there. All right, so I'm going to stop this video here, and then I'll do another one where I go through that lac operon worksheet.